Hello, my name is Terrence Barkin. I'm the Executive Director of the Graphene Council, and today we're hosting an interview and session with Dr. Kiana Aran from Cardea. We are going to learn a lot about this exciting technology, especially for the application in biosensors. And with that, Kiana, it's all yours. We're very interested to hear what you have to share with us today.
Thank you so much. I'm very excited to be speaking on this platform and for the Graphene Council. It's a pleasure. So I'm the CSO and co-founder of one of the co-founders of Cardea Bio. I'm also an associate professor of bioengineering and medical devices at Keck Graduate Institute. So at Cardea, most of you know, at Cardea, what we basically make graphene transistors, and we make a fully functional graphene biosensors. But before I go and dive deep into how we make this sensor and what applications we have, I want to walk you through what a transistor is and where we are today in terms of basically graphene transistor technology. So in early 1900s, many of you might remember vacuum tubes. So vacuum tubes were basically switches that were used for applications such as long-distance telecommunication. They were bulky. We also had vacuum tubes in radios. These vacuum tubes were bulky. They needed continuous replacement because they would break very often. So around 1950s, AT&T commissioned Bell Lab to develop better switches that they could actually do long-distance communication without having to maintain them so often. So this is when William Shockley and his colleagues at Bell Lab developed transistors by switching from vacuum to germanium and silicon. And this was the beginning of the silicon era for us, where we had faster switches, faster transistors, smaller transistors, which allowed us to really have more compact system today. So basically in every gadget that you hold in your hand, every electronic gadget that you have in your hand, there's a transistor component in it. And at the time, no one knew the application, the wide and the vast variety of application that this transistor would enable us to have. Later on, in around 1960s, we developed integrated circuits made from these transistors. And the reason for that was that you could not give a person a transistor and ask them to use it. No one would know how to use it. So in 1960s, we developed integrated circuits that basically had more capabilities. They had elements of transistors with other element, memories, other component, electronic component, which really enabled us to have CPUs, GPUs. And today you can buy a CPU and a GPU, and basically you can build your own computer out of it. So basically transistors themselves, could not leave an impact if they were not, incorporated in those integrated circuits. Both transistors and integrated circuit IC basically won the Nobel Prize in 1956 and 2000 because they were really fundamental innovations that enabled us to be in a digital age that we are today. So it's amazing. We have all of these gadgets, all of this electronic that we can use for many applications, but it would have been great if we could use this gadget to connect with our health, to connect with our own biology. And this is why at Cardea, we wanted to actually move on to really create transistors that could actually speak with biology. Silicon is great, but when you want to, talk with biology, silicon material is impacted by salt, by biological material, and if they loses their sensitivity, they also impact biology. So they won't allow biology to do its function. So we're looking for that next generation material that enable us to speak with biology at the speed of biological interaction. So we used graphene. Graphene was a 2D material which was discovered in early 2000, won the Nobel Prize in 2010, and we have a lot of application from transistor in different industries. For us, we wanted to make this next generation transistors that could connect with biology using graphene material because of all the superior characteristics that they have compared to silicon. So we decided to really make a full system and build more than just a chip, more than just a transistor, but also a full platform that enabled people to connect with biology. Basically, we wanted to create this fully functional biosignal processing unit called BPU that can create a gateway between the live biosignals and the digital world. And we really need this, with all of these advances in electronic system, we really need to have a platform that can enable us to talk with biology, to be able to connect with our own health and biology. So just to tell you how do we build this system, this is the picture you see on the left is actually a picture of a BPU. If you zoom in, you see many black rectangles. Each of these rectangles are a graphene transistor. When you zoom in each of these graphene transistors, you see that we have conjugated a certain biology to our graphene surface. So each of these transistors have three terminals, two terminals, where basically there is a graphene layer between the two terminals, namely source and drain. And then there is a third terminal called gate that is immersed in the solution. We actually anchor different type of biology to our graphene sensors, and we look at the interaction of this biology with our analyte of interest that is found in our blood, in our serum, in any biological sample. And as this binding happens between the anchored biology to our surface and the actual analyte that we're looking for, we can see the signal in a live with our sensor, because these sensors are extremely fast in monitoring and seeing this electrical signal. And the beauty of biology is everything in our body is charged. Our proteins are charged molecule, our DNA is a charged molecule, RNAs are charged. So as this component, anchor and bind to our graphene transistors, we can monitor them in real time. And this is basically the core of how the technology works. This is amazing, and the amazing properties that using graphene can allow us to do, it's really to harness the power of biology itself. What I want to talk about here, it's if we get to actually build this system, a fully functional system, we can do things that no one else was able to do. For example, what we did, we anchored a CRISPR molecule to our graphene, to our BPUs. And the beauty of that was that we were able to watch as CRISPR was doing its function in real time. So CRISPR is this two-component system that was found in bacteria. The two components work together to basically defend bacteria against viruses. How it works, it's a nuclease enzyme that can target the DNA or the RNA at a specific sequences that is programmed by an RNA molecule. So basically the RNA molecule will tell the nucleus where to go and where to cut. It's so powerful that it can unzip double-stranded DNA in a cell, look for its target, finds it, cut it, and then, today humans are using it for editing applications as well. But the beauty of it, when we look at this powerful biology or nature-created technology, we wonder, can we actually use this and take advantage of that? And with graphene sensors and graphene biosensors, we can actually do that because of the speed and the biocompatibility of this system. So here we were able to actually anchor a CRISPR and watch it as it finds its target. The potential, the power of this technology was that normally the way that we find a DNA, we have to take a DNA, chop it, then amplify it, then add label to it, then we'll be able to amplify it, then we'll be able to detect it. Using biology as technology with the power of graphene transistors, we were able to actually eliminate all those steps and take a whole genome, basically a buccal swab, a swab from the mouth or the nose, and put this genomic material on the transistors and allow CRISPR to do what it does in nature. And this is fascinating. And when you think about it, CRISPR is not the only molecule that have a very fundamental function and amazing function in our body. There are other molecules. For example, there are olfactory receptors that kind of smell. You can imagine anchoring them to transistors and watching as those olfactory receptors, those proteins basically find the molecules, could be a smell of something or a small molecules, and we can develop electronic notes, for example. So there's a lot of power to being able to really use this amazing material, graphene, and be able to connect it to biology, to be able to harness the power of biology. And also because our platform does not require optics, does not require labeling. You can, with the same platform, by changing the biology that you anchor to the transistors, you can detect different things, different analytes. You can detect biomarkers that are associated with cancers, like free-circulating DNA. You can monitor exosomes, which are these extracellular vesicles that are carrying a lot of information that are used for cancer research and other pathologies. You can look at immune response. You can look at circulating cells, and you can look at volatile organic chemistry and a lot of different analytes within the same platform. And this is really the future. In the digital age that we are moving forward to, we cannot send our DNA sample to one lab using one instrument, send another sample to do RNA analysis to another lab, the protein to different lab. We need to actually have a platform that can monitor all of these analytes on the same platform, because this will enable us to tap into biology and look at these markers at the same time, which then enable us to really understand biology much better. So since I'm the chief science officer, I'd like to dive into a bit more about the science that we have been able to achieve with this amazing graphene biosensors and BPUs. This is an example of using these sensors, using these graphene biosensors to detect mutation in our gene without amplification. So what we did here, as I mentioned prior, we anchored a CRISPR molecule that was programmed to detect a specific mutation in gene obtained from patients with Duchenne muscular dystrophy disorder. So this disease impacts 79 exons in the dystrophin gene. And any mutation in this exon could lead to this genetic disease. So we're able to actually design a sensor that could identify the presence or absence of a mutation in the dystrophin gene. What you see the middle plot here, it shows you a CRISPR chip, basically a graphene-based biosensors functionalized and powered by CRISPR, where we were able to detect the presence of a gene, which was a gene exon 51 in healthy sample, H1, H2, H3, and sample A, B, and C, which were also diseased, but they had that specific exon, and sample D, E, and F, where they were lacking that exon. And the chip was basically able to identify the presence or absence of those sequences without amplification. This is just taking a buccal swab, putting it on the chip, and monitoring to see if that specific sequence was present or absent. This is super powerful, because you can imagine having a single chip with hundreds of these transistors. Each of them are programmed to detect a specific sequence in the genome. And then this way you can identify the presence or absence of a lot of known mutations that are associated with diseases without the need for amplification, without the need for all of those reagents at centralized lab. And this is the power that this graphene transistor can bring to the market. We can also quantify that. So not only detect them yes or no, which which could potentially be done by PCR, but we can also tell you how much of it is present. And this is super, super powerful. The next thing we have done, and both of these studies are published in Nature journal, we've also tapped into more powerful biology to see if we could do more amazing things by connecting them to our graphene biosensor. So what we did, we used a CRISPR enzyme that was more specific to detecting single point mutations in our genome. So single point mutations are basically when only one base or one letter in our genome has been altered or modified, and this can cause to about 50% of genetic diseases. And so example of those genetic diseases are sickle cell disease, ALS, and other type of diseases. So using this specific CRISPR enzyme that was very sensitive to single point mutations, we were able to actually detect a single letter alteration in the genome without amplification in sickle cell disease and in ALS disease. Not only we did that, we were able to detect the zygosity of those mutations. That means, are they present in one allele or in the other allele? And this is amazing because there is no technology out there that could do that without amplification, even with challenges, if ever possible, to do such things. So this is example. This is again another example of the power of this graphene transistor and what can be achieved with this transistor, tapping into the power of biology and harnessing the power of biology. We were also able to detect exosomes. So what you see here, this is another work that's been published, that we were able to detect the presence or absence of exosomes. These are extracellular vesicles, basically about 50 to 150 nanometers, that contain RNA and protein molecules, and they are the major component for cell signaling. So this is how cancer metastasizes. This is how cells are communicating with each other, and a lot of diseases are basically spread throughout our body. They're super important in biology these days. We were able to basically attach an antibody that was recognizing a surface receptor on these exosomes and monitor them, monitor the concentration of these samples in human blood and in mouse blood and in age population and younger population, and see their changes. And the amazing thing that we've achieved a limit of detection that was significantly superior to ELISA, commercial ELISAs. In commercial ELISAs, you can get an LOD of 10 to the 9. In our sensors, you could get an LOD of 10 to the 5 exosome per ml, which is really important because sometimes you want to detect those earlier during the disease, so you can diagnose diseases faster. So I gave you a few examples of some of the amazing assays and biology that we have been able to detect using this graphene transistor. But I want to also walk you through what it takes to actually develop a sensor like that. So let's say a simple, which is not very commercially relevant, an ELISA assay. And I'm talking about ELISA because it's the simplest one I can walk you through. But really the power of our technology is not only ELISA, because there are a lot of commercial ELISAs available. And if you're only focusing on ELISA, then you're not really tapping into the power of this graphene-based transistors. But I also want to emphasize the importance of multiple different components and knowledge that goes into developing and is required for developing these sensors. So what you see here, I'm showing the 4 different components and elements that goes into developing a fully functional platform. So the BPU, basically the graphene transistors, is only one step of it. And you have the biology and the chemistry to actually anchor the biology to the graphene surface. You need the hardware to actually develop and read the signals from these transistors. You also need the software and signal processing and signal analysis, which is very, very important to understand for understanding like what is the signal that you're getting from this sensor means. So the BPU itself, we call it the nanoware itself, requires a lot of attention in terms of graphene standards, quality control, being able to reproduce the material. And I think my co-founder and colleagues, Brett, have a video which he walks you through all the importance of this, to have a transistor that is reproducible. And that's the first step of the process, where you want to make sure that all of these transistors are responding the same to the liquid or to the buffers or biological component that is introduced to them. So that's very, very important. The next thing is anchoring the biology to the surface of the graphene. That requires extensive knowledge of surface chemistry. How does that surface chemistry impact the response of these transistors? Because these transistors are super sensitive to everything that is bound to them. So you need to understand, what are you doing with the transistor, how to understand those signals. You need a great understanding of physics, electronic engineering, surface conjugation, surface chemistry, and biology and molecular biology to understand and be able to reproduce this system. Because if you don't understand what is the impact of changing the buffer on graphene, or what is the impact of a specific charged molecule with a specific isoelectric point on the transistor is, then you won't be able to analyze the data. Because when you're actually doing the data analysis and you're reading the signal, which itself requires understanding what different reader components you need, what parameters you want to extract from these readers. And really, there's a lot of knowledge goes into the hardware. But also, if you don't understand these behaviors, if you don't understand, how much of this signal is really associated with your actual biology, then you won't be able to develop signal processing and formulation that can extract the actual signal. So we have done at Cardea, we have worked over 8 years with biophysicists, chemists, engineers to basically understand all of this aspect through design, experiment, repeating with different assay, and really understanding if you're putting an antibody, what sort of a response you're expecting to get, which electrical parameter you need to measure, what combination of electrical parameter you need to measure, and how does that differ when you're actually measuring an exosome or you're measuring a DNA. So there's a lot of knowledge that goes into really understanding this. And once you get that, then you really can tap into the power of these graphene biosensors. So again, we offer the whole package. We offer, at Cardea over the 8 years that we have been working on this, we have developed not only the reproducible transistor, but also the hardware, the software, the knowledge of how do we actually bring different assay and make them an electronic assay. And this all allows us today to offer our BPUs to people and partners who wants to utilize our platform, because we have made it super easy. You don't have to have multiple different readers and amplifier the same way I did when I was a postdoc to connect this. I don't have to look at different signal to see, okay, what does the signal means? All of these have been created for our partners to be able to take those, just decide of what biology they want to, basically detect. We will help them to design the wetware, because we have extensive knowledge on that, and the signal processing that will be done based on the biology that they're interested in, and they can get the result in a plot, basically using our softwares. And I want to get to also how everyone can basically get access to our technology. We have a program called, this is our innovation partnership program, that people can contact us, schedule a meeting with us, talk about their needs. And we have a standard licensing and revenue sharing models that we can, talk with our partners about. And we basically develop and help them develop a product using our platform. And we have a dedicated support team to basically enable people to basically get access to our platform. If you want to know what else we can do with our platform, we have a whole bunch of publication through our company and our, people who have used our platform and, obtain some results that are listed here. And if you just go to our website, you can find more of these publications. And again, we are offering the full package. You don't have to figure out, okay, now I have a transistor. How do I connect the wires to it? How do I read from it? How do I analyze the data? We really provide people with the full platform to be able to use our technology. And I just want to go back again here, talk about and emphasize, we are here because we have spent many, many years getting the sensor, that the graphene transistors themselves and manufacture them consistently and in a reproducible manner. What you see on the bottom graph here is basically it shows you that the tight distribution of electrical parameter that we obtained from our scaled transistors. Back again when I was a postdoc, I would make 100 transistors, and I would get 10 of them working. And throughout these years, and my colleagues, that have spent many years to just understand how to manufacture this so we get more reproducible transistors in a way that they are ready to be used with biology. Now, so that's the first step. The second step is we require a lot of understanding of the chemistry that goes on this chip. What sort of a chemistry you need to use without impacting the sensitivity of these transistors. How do we make sure that you always get consistent and reproducible result? Again, we have done significant amount of work, with the biochemists and physicists and surface conjugation chemists that have helped us, throughout these years to basically understand this better. So now you functionalize and you attach a certain biology to your transistor, then you need to understand the next step. How do you consistently get a bioassay to work and understand it? Because enzymes can sometimes work differently. What are the quality control metrics you have to have for your biology? What are the quality control metrics you have to have for your transistors? All of this needs to be in place. And the workflow that we have designed really tap into all of these aspects to be able to provide a signal that is meaningful, and again, it's beyond just creating a transistor. It's beyond just knowing the assay, the biology assay, because there's a lot of knowledge, of conjugating this biology and electronic circuit that is required to be able to do that and give a reliable signal for the assay that we're developing. So with that, I want to conclude that if you're really interested, we have our Francie Barron. She's our VP of Innovation Partnership. She's an amazing person to talk with you guys and walk you through what your needs are for your assay. And we would love to collaborate, and we would love to have people use our technology. Again, I want to go back to the transistor era. Creating a transistor was not enough. Providing a solution with the transistor was what actually led us to where we are today in terms of technology. We want to give our technology to people in a solution, and we want to develop solution with them, because this is how we can really bring the power of graphene transistors to the market, bring them to the hands of people that can really benefit from them. And this is our goal at Cardea. When we have partners, we have a lot of partners that are working with us, and these are industrial partners, Siemens, Gate Foundation, CRISPR QC. We're working with DARPA, with GTRI. We have a lot of academic partners from Berkeley, Stanford, Bar-Ilan, and there are many academic partners that have used our platform and have published with our platform. And we're looking forward to having more partners, because again, we want to bring solution, not just a transistor, but the full package of solution to our partners to enable them to do what their interests are. So with that, I want to finish my talk, and let's see if you have any question for me.
Well, Kiana, thank you so much. That was absolutely fascinating. And I think it takes a little while to absorb just how important this advancement is that you guys have created. I have a couple of questions for you. So one of the questions, when we take a look at the actual chip itself, there are multiple points on there that have the graphene transistors where you can put the analytes on for testing. Is it foreseen that you put multiple analytes on one chip, or is it simply more detection sites? Can you talk a little bit about that?
Yes, absolutely. So in this version of our chip, which have taken us a long time to develop, we just wanted to make sure that every chip will give a response, or you get some sort of response from every chip. And if for any reason one of those transistors were broken, you have more transistors to actually test. So it's mainly for getting more basically ends per chip. So each chip now gives you N of 3. There are 3 circuits in each chip, and each circuit has 3 channels of graphene. So in case one of them break, you still have 4 other channels to test. So it's just ensures you, and this is all from a marketing perspective, we want to ensure that everyone can actually get a result from the chip. And if for any reason you broke one chip, you don't lose all of your result. Having said that, the future design, which are in development, are designed in a way that you can actually, have 4 different type of biomarkers in the same chip. And they are designed in a way that it's very easy to functionalize them separately using commercial systems. So we are moving towards that, and the future will be many more transistors. And I think the importance of it is that we don't want to give something that has hundreds of transistors without actually knowing what the application is, because sometimes it's marketing. You don't want to create something that is potentially harder to scale without knowing what is that the customer wants, right? By ensuring that we have a reproducible system from the chip production, from the assay, understanding the assay and conjugation, and knowing what are the application of it, then we move to 4-plex, 16-plex, 32-plex, 1,000-plex, and this will come after that. Understood.
So in short, you have some built-in redundancy into the chip at the moment, and then you have the possibility for that platform to extend to do multi-analyte analysis, right, as you go. Um,
Absolutely.
I think,
There's one more point I wanted to add here before we forget. There is also, with the same chip that we have right now, you can still do multi-analyte, but it really depends on application. For example, if you're trying to detect the presence or absence of 4 specific genes, and you don't care which one is present, because any of them, the presence of any of them could lead, is an indicative of a disease, for example, then you can actually put the mixture of the capture molecule on the same chip. And then if you get a yes signal, it's there. So it depends on the application of how you want to look at it.
understood. I think that speaks to the next question I would have for you, and that is, what is really the limitation on the types of biology that you can detect with this? Because it sounds like, well, let me say it this way. What I think is absolutely amazing is you have this consistent platform in terms of both, the chip itself, the analytical tool, the software part of it, and so you have a consistent platform, and you can detect different types of biology. There are some that are obviously going to be your first targets, like the example you gave with CRISPR. And I guess the question is there a limit on the types of biology that this tool eventually could be used to detect?
I personally think that there is no limit, but there is one, and that's your knowledge, right? Because when you're conjugating an antibody, there is a different signal that you're measuring. When you're measuring an exosome, it's bigger. The charge is bigger. Then you have to think about different signals that are combined together. And we have developed that knowledge throughout working ourselves or working with partners based on the analyte that they need. So it's important that we actually test all of those analytes to understand the biophysics, to understand the electronic property, to understand what signal needs to be processed, what signals are better for exosome, what signals are better for DNA or RNA, and which ones are required a combination of this signal. And I think it requires a lot of testing. It requires working with partners who understand that biology and developing those assays. So right now we have done that for exosomes, proteins, DNA, some of the small molecules. We're working on volatile organic chemistry with the Gates Foundation. So as a company, we have to work with partners to actually add to this modules, let's call them this way. And I think, but to me, it's just that understanding what needs to be measured, measuring them, putting biophysicists, chemists, electrical engineers together to sit down, and bioinformatician to sit down and understand those signals. And then we will have a full library of signals and bioanalysis that we can detect, and they all can be detected, yes.
So that does point out the other important topic here that I don't think is quite so obvious, and that's the fact that you guys over the past 8 years have accumulated this body of knowledge through all the trials and testing that you've done to build that library. Because as far as I know, that's not something that you can go and reference. It's not something that you can infer. It's only something that comes from actually testing each of these different biological signals over many, many, many reputations to refine what that signal looks like. Am I missing something? There's no shortcut to that, is there?
Absolutely not. I'll give you an example. For example, with our CRISPR chip, we have tests of 500 transistors to actually compare the result, analyze, understand, and develop a software that can actually detect that. And and our knowledge, as we expanded our partnership with different academic and industry partner, and brought people from different disciplines together, was what really enabled us to get the ones that we already feel comfortable with working. And we're really hoping that if there are other markets, for example, with volatile organic chemistry, with our collaboration with Gate, an expert in transmembrane proteins, which can detect the VOCs, we can understand how does the biology is impacted by electronic, and all of those things. It's really important to understand and develop those assays. So it takes a lot of effort and a lot of people from different disciplines to get to a point where we are today in terms of those set of analytes that we can detect. Yes.
So if you don't mind, I have some practical questions that kind of cut from the biology straight to the business case here, and that I can imagine there are thousands of people working in labs just thinking, wow, I can't wait to get my hands on this tool, but they're not always the decision makers on what analytical tools are used. And you have obviously embedded practices and equipment and this sort of thing to overcome. Who do you see as the primary decision maker to have a look at this tool and say, yeah, this is something we want to trial in our labs, or this is something that's going to allow us to do something we could never do before, but would be a tremendous help to us, especially in this healthcare field?
Yeah, absolutely. So it requires awareness, and awareness comes from collaborating with people and showing them what capability of the technology is. So early on, we had collaborated with a lot of people with basically even not much financial gain to just bring awareness to what can be done with this system. And that led to further continuing our collaboration and them buying chips from us. And now they're using our instrument. You know, a very good example of that was it was a faculty in Stanford that moved to, Bar Ilan University, and he bought our platform. He's using our chips, and then he's expanding that to other universities in his country to actually use our system. And we are collaborating with some cancer institute now to let them, use our platform. So it requires bringing awareness. It's not, people are not aware of this platform. So talking at conferences, showing what we have done with the technologies, showing the capability of the technologies. We were able to actually have a spin-off company, CRISPR QC, which offers solution of really monitoring how a guide bind to Cas, which is not, this was a great application because it was not possible with any other instrument. So for me, it's, I think, bringing awareness, giving talks to universities, showing them, don't just give them a transistor because they can't use it. I used to buy, graphene chips when I was a postdoc, and I had to actually make them go to nanofab, pattern them, connect electrons. This is very difficult to people to use. So in order for us in graphene industry to really expand the utilization of these technologies, we have to offer this full solution to a biologist, where when he compares his normal standard assay with our assay, he sees that, okay, it cuts down a day from his workload, right? So we have to show this. And I think the first step is offering full solution, not just a graphene transistor, but also, again, the full package, and showing them how much of their workflow can be reduced and how much the cost can be overall reduced, because you're cutting on FTEs, you're cutting on reagent, and you're getting to more results using the same platform. So I think this will come from bringing awareness, collaboration, offering, and the major one is offering full solution, not just something that, you leave it up to the user to figure out how to use it.
Absolutely. And you've anticipated some of the questions I might have had for you, and that's on kind of the business case where, they could, as you mentioned, so a customer could take the chip, take the equipment for the interpretation, they take it to their lab, they do their own testing, they accumulate their own knowledge with it. And it sounds like with the subsidiary spin out, is that because there's so many analytical labs today that simply offer the test results as a service, and nobody knows what happens, behind the scenes, so to say. And I could really imagine, especially because this opens up such a broad panorama of detection and analytics that you can do with the same platform, that I could imagine quite substantial businesses built on delivering these analytical services as a service. So I don't know how far along that is, but I imagine there's some partners you might be discussing that would be considering that, and if they're not, there should be.
Yes.
So do you want to speak to that a little bit?
Yes, you're absolutely right. So we are speaking with some partners that could utilize our platform. And these are really partners that they see the vision, because again, if you have a partner that just wants to do ELISA, then they're like, okay, you have to match all of these ELISA LODs and you do all of this thing. And then you go like, okay, what is the commercial benefit of that? There's a lot of ELISAs available. They get cheaper and cheaper. The power of our technology comes when you think about the multi-omics aspect of it. There is a whole bunch of research in cancer, in aging, that is showing that looking at one analyte is not enough. Right now, despite the whole, plethora of knowledge that we have about the importance of monitoring this analyte, because the instruments are not available, and because the doctor have to send the DNA for sequencing to one lab, the RNA to sequencing to lab, they don't use that knowledge, And I think the best way for us to, and we are doing that, to work with people who knows where the future will be in this sort of diagnostic and this sort of testing, because I think this sort of transistor, graphene-based transistor, will be the front line we'll be at the front line of this technology, because these are the only platform that you can do multi-omics without the requirement of labeling. So understanding the power of them, it's important for people who understand not just another ELISA, but can we actually, what else we can do and bring with a single platform. And for us, yes, we are, to answer your question directly, we are talking with a lot of partners and who are interested in this multi-analyte aspect of our transistors.
Another aspect along those lines that really did catch my attention was the sensitivity, right? So you could do an earlier detection, or you could have a more sensitive testing. There's a theme that's been important to me for decades, and that's the concept of community health, and that's the ability to really analyze and test for a whole society in a preventative way. Preventative medicine obviously doesn't get nearly enough attention, and even with the recent pandemic, that should raise people's awareness that we really need to be vigilant. And it would seem to me that having multi-analyte testing ability, do it fast, higher sensitivity, and quick is a perfect tool to think about doing more preventative medicine. So that's something else to think about. My question for you, though, is on not just the sensitivity, but false positives, because every test method that exists out there does have a degree of uncertainty to it and does provide false positives. Do you have any data about your technology in that area?
Yes, so that's a very good question. So the importance of designing the right control comes from making sure you talk with the right people. Because if you only have, a certain set of people like me or a physicist and someone who's developing the sensor trying to come up with a control that always shows you the false positive, it's hard. So you need to work with, oncologists, cancer expert, to technology developer for those specific applications, see what sort of control sample is acceptable to them. For example, when we work with sensors from Department of Defense, they wanted us to have a very specific control, which was, a specific analyte that they would detect as a negative control. So we have to adapt our system to basically detect the threshold, the signal that we get from that non-specific one. And for each, when you're detecting DNA, that control is very different. So it's important. Again, that goes back to the importance of, understanding the biology also and the assay development aspect of what you're developing, because the background, the threshold that you're getting from those analyte will be basically the noise level of your system that you need to understand and incorporate in your signal analysis. So, and that's one aspect. And also for us, another signal that we needed to really understand is the behavior of different buffers and how they react with our sensors, and create, basically a library of different buffers that work the best with our platform. If our customers were interested in other buffers, we actually have ways to examine those and understand their behavior. So these are really important. And this is why, when I see a publication coming out with 3 sensors, because they saw some results, and some separation without the right control, without the right understanding of how this biology will be basically interpreted when you're scaling it up, it hurts me as a scientist, because I'm like, come work with us. We have spent all of these years developing these things. We want to move the field forward. If you really want to do an assay, come use our knowledge. Come, let us help you. And I think this is really important. So the understanding of what are the best control to understand the threshold signal also comes from years of experience and working with partners and collaborators.
Well, that raises another really important point. And my question would be, how do you capture these the knowledge and experience that some of your customers are creating? Is that shared? Because it would seem to me like maintaining a larger and larger pool of data is to everyone's benefit.
Yes. So this was actually the business model that our CEO created, which I think was an amazing model, because the way we wanted to expand our modules, and when I say modules is understanding the behavior of the sensor with different biologies, no company alone could potentially do all of that research. It's a big research. So our model is that every company who partners with us, it's actually benefiting the next company who's partnering because they add to our knowledge. So throughout this multiple collaboration with industry and academia, we have gained that knowledge. And for some proteins, for example, we realized that wow, the same analysis can be used. And then we look back and like, what is the similarity between these proteins? What is their behavior in different buffers? And this is the understanding that I'm talking about. Because then when the next protein comes, we look at it to see how similar is that protein to the assay that we have developed, or how different it is. So this way it allows us to be very confident in the signal that we're giving our partners. So again, it comes from really extensive collaboration, running those assays and years of knowledge, and speaking and putting the minds of physicists, biochemists, surface chemists, conjugation chemists, molecular biologists, engineers, electrical engineers together to be able to really find a solution.
Well, you started the presentation with the analogy of the evolution of electronics and the ability to detect and make transistors, and how it becomes more and more and more sophisticated. And the way I interpret that, or my observation, has been, we constantly move forward to attack the next higher value problem, right? Instead of doing rudimentary things way in the back. And it would seem to me that what you and your teammates have created is a step change in moving to the next level, moving beyond optical type of sensing, which is common in the analyte or in the analytical and diagnostic field, and moving more to these electrical signals, and really leveraging graphene, which until this material was available for this type of detection with these very specific electrical signals, extremely high sensitivity, and the ability to use it directly in connection with biology, because it is biocompatible, pure carbon. It just didn't exist before. And so we're really at the front stages, and already you've accomplished so much, and it's been, in the grand scheme of things, actually not that much time. It's exciting to think about what's coming in the near future. From my side, I don't really have any other questions for you. I think this has been very enlightening. I think you should be extremely proud of what you've built, because it's not just one thing, it's a whole family of capabilities that you have connected together in a meaningful way, in an elegant way, I would say. And we're also very happy to have you as a member of the Graphene Council. It's exciting to see graphene being used for such a high value proposition.
Thank you so much for having me. I just want to end with last thing. Let's not forget that, again, back to the story of transistors, when Moore was making faster and smaller transistors, the Japanese industry were actually using the older version to make useful systems. I think this is the mentality we need to have. We can actually offer solution with whatever we have in our hands today, to bring awareness, to introduce what this amazing platform and technology can do, and slowly improve it with the help of the whole ecosystem of people that we have in Graphene Council, partners, academic partners. And this is how we can, it's our goal to bring this technology to the world, and this is how we can actually advance. And I hope that we all can work together to get to that point.
Thank you, Kiana. So for those of you who are interested to learn more, please reach out to Cardea directly, and they'll be happy to give you more information about what they're doing and engage with you. If you're interested simply to learn about graphene in general, the Graphene Council is a neutral global community that gives independent information and analysis of graphene, both from the research side, graphene production, and end use applications. We're here to help spread the word and to provide education and information on this really important technology. So from our side, again, thank you so much, Kiana, and appreciate your time and your expertise.
Thank you so much for having me.